I would like to offer you the outcomes of an experimental application of Gaia in relation to MF, as was said, and in a pre-fermentation setting, which is considered here in relation to the drying of grapes. So the drying of grapes is a practice whereby you transform the grapes, the fresh grapes, and that is used in several situations, in several wine-producing areas in Italy. It boasts a very ancient history, and it can be done in various ways. As you see here, in the pictures. In one case, the grapes are exposed to sun radiation, which means that concentration happens rather quickly. Or in other cases, and this is more typical in the north of Italy, you harvest the grapes and you take the grapes to protected lofts where the grapes can be kept for a long time. And they are dried there within the framework of a slower process. The case we considered is the specific case of grapes used for the production of Ricciotto and Amarone. And we know that in this case, the drying technique is a technique that has become very popular, and Amarone has become very popular as a wine, hence the increase in this type of approach. The grapes used for this wine are kept in the drying lofts in a protected environment for a rather long time, for as many as 3 to 4 months. And they are red berries which are put on natural mats and or boxes that can also be made of plastic, as you see here, and they can, in this case, be handled more easily. This also leads to this type of situation where the drying lofts are built specifically for the stocking of the grapes, and they can accommodate huge volumes of grapes that, of course, need to be handled very, very carefully and managed very carefully. As I said, grapes remain in the lofts for rather long periods. The management of grapes during that period is a management that can entail problems. Some of them are very well known and have been problems since the very beginning, and other problems have emerged more recently in relation to new techniques. Because you see, if you work with a small quantity of grapes in a small environment, you can, for instance, select the grapes during this drying process. But this does not apply to the case when you have huge volumes, as is the case here, bottom right picture, where these grapes are placed in one specific place in the drying loft, and they remain there. The management of huge amounts of grapes also entails that you apply the right conditions in the drying loft, and that is key in order to have a uniform dehydration process, and the kinetics too of this process has to be considered, which is what I would like to talk about, because one of the criticalities that emerge increasingly is linked to the rapidity of the drying of the grapes in a drying loft. Here you have the speed of the reduction of weight of the grapes in the same drying loft, 3 varieties though, in 3 years. And the in blue, you have 1 year whereby speed was much faster, so much so that grapes were ready to be vinified basically in half the time of the usual speed which you see in the other 2 cases. Why is this a problem? It is because we know that the dehydration velocity has to be slow enough to make it possible for the grapes to take on those specific dried properties that then you find into the wine. If dehydration, if drying happens too fast, that is detrimental. The point is, therefore, the way by which we can go back to traditionally slow drying process. Well, you can work on the climate conditions in the drying loft, but that is a difficult and delicate matter. The climate conditions have to be favorable, and you have to consider, though, that if something goes wrong, what happens is that you might have rotting berries, and rot is a problem that we know also from the past. You have these clusters of grapes which have these foci of botrytis and mold. This is a picture from 2016, so it is a very recent situation. And the experimentation is also recent. It was done last year, and the experiment was that of using MF, Methicillin fructicola, which is Gaia, in order to inhibit the development of botrytis. The experiment that we did involved huge batches of grapes, so much so that we could go for microvinifications. The climate conditions adopted were special in order to induce a strong botrytis aggression. We did not inoculate botrytis artificially, but we created the climate conditions, the environmental conditions, so that the natural inoculation which is present on grapes would develop more aggressively. And based on that, we assessed the capability of MF to control the molds. Actually, we did 2 tests. One had to be stopped after 25 days only, and there we had 2 dosages of the Gaia product, 20 and 50 grams per quintal. And then the second test that started immediately afterwards, which grapes that arrived at the drying loft at the end of October, lasted sufficiently long, and that made it possible for us to monitor a drying process which would be similar to the normal one, to the real one. And here we used only the higher dosage, that is 50 grams per quintal. This test made it possible to vinify. And here we have some preliminary data. This is a very recent analysis, actually. But anyway, we already have data to offer based on these recent outcomes. The variety is Corvina Veronese, which was put on this plastic crate. And it was not placed in the normal drying lofts, but in artificial ones, smaller, where we could control humidity and temperature. throughout the drying process. Here we see a picture of the drying loft, so to say, of the artificial drying loft. Here you see that humidity is in blue, and in the very first days, the humidity levels applied exceeded 90%, so very high levels for a very short time period, 3 to 4 days, which made the natural inoculation of botrytis on the grapes develop massively. This has to be then checked visually. Humidity was brought down to more typical levels of a drying loft.
Per quanto riguarda la
As far as temperature is concerned, declining temperature levels were reproduced in order to mimic the normal temperature in a drying loft, which is generally not controlled, but it is, there are natural fluctuations based on outdoor temperature. It starts declining between the end of September to the end of the fall season of the autumn season. Also in the second sample, we reproduced the same conditions, both parameters high at the beginning and then declining. The drying kinetics. This is an important element in order to understand how drying is going on, how well. There were no major differences, we can say, between the grapes that were not treated with the product and those that were treated with Metchinkovia. What we saw was that probably towards the end of the test, the untreated grapes, or rather the grapes, the speaker is correcting himself, the grapes treated with the highest dose of Metchinkova tried to sort of slowed it down in terms of dehydration, which might indicate a less extensive presence of botrytis, because botrytis does speed up drying because it causes cracks in the skins of the berries that favor the loss of water. In this case, but the test should have been extended to see that more clearly, this might have been a symptom for the for the prevalence of botrytis on untreated or more treated but at lower doses grapes. Now, the next section is the one that my colleague Fracchetti should have dealt with. Unfortunately, he could not attend for health reasons. Now, to really understand whether the grapes could be differentiated based on the fact that they had been or hadn't been treated, and therefore based on the spread of botrytis, several analyses were carried out on the grapes. Now, we asked ourselves the question about the possible action of these yeasts, and what the mechanism of action might be. This is something which is still much of an assumption because we have no certainties in that respect. But apparently, it could be an action that occurs by subtraction of nutrients for other species. So that might happen through a chelating agent, pulcherima or pulcherimine, which is a chelating agent that particularly attracts ions, which might benefit other species that might thrive on it. This still has to be more extensively tested, but as my colleague shows here, there are many examples that seem to point to the fact that Metchinkova fruticola is an antagonist to different species, different other species, including botrytis. This was tested in vitro, but also in fruit, like the apples I showed at the beginning, other fruit during storage, but of course in vitro testing can be very different from what happens on the surface, on the skin of the fruit. And in any case, storage of fruit can be very different from the drying of grapes. So it's not a matter of preserving the initial condition of the fruit. So a test on drying grapes is something that is unprecedented, and it assesses an aspect which had been unassessed so far. Also testing the microbiota that naturally lives on the grapes during drying, and that can even be detrimental to the process and the quality of the grapes. The test was carried out by spraying the grapes manually of a suspension of the product. We used a sprayer on individual crates. The crates were allowed to dry for a short period of time, just a couple of hours, before being placed inside these chambers that mimic the drying lofts. We also introduced some traps to avoid the proliferation of certain insects, which are another scourge for drying grapes, such as the moths and flies that could also spread contaminants, other microbial species from crate to crate. The yeast viability was confirmed using molecular methods based on the DNA, and also fungal attacks were confirmed using DNA isolates on plates. At the end of the process, the samples were vinified, were crushed, and the results were pretty encouraging. Now, after spraying, this is what the grapes looked like. They were wet. They were allowed to dry before being placed in the drying lofts or drying chambers. At the end of the test, you still had these white spots because of the yeast suspended in the sprayed fluid. It's not mold. And even in the next pictures, do not confuse, do not mistake the spots for mold, because it's not. It's two very different things. And also the molds, if the grapes are moldy, you can tell because you have a focus from which the mold spreads. Now, when it comes to the viability, the result was pretty surprising. Once the grapes had been sprayed with this suspended Metchinkovia, and after it had been allowed some time to dry and then placed in the drying chambers and then dried again, when you actually went back to the grapes, assessing the possibility to still find a viable Metchinkova on the grapes, it was still there and viable. Throughout the period at 45 days, the Metchinkovia were still viable. Of course, you found more for the highest doses in the solution, in the suspension, but it was still there also in the other samples. This is an indication that this yeast can have an action also down the line during the drying period, also at an advanced stage, pretty late. This is the molecular test assay that confirmed the viability of the Metchinkovia in the sample taken from the grapes. You see the recognition of the Metchinkovia in line 6. In line 7, the assay did not recognize any other strains of Metchinkovia, so this is a discriminating assay for that too. This is very useful information because it makes it possible to then carry out other assessments to see if the product what you find in the grapes is the product that you've actually used to spray them. A typical way to see whether the grapes were or were not protected from botrytis is the visual analysis of the berries. The visual analysis makes it possible by just simply looking at the berries and classifying them based on whether they are healthy or moldy to measure the prevalence of the mold on the grapes. In both experiments, you see that untreated grapes were attacked by botrytis with a proportion of moldy berries much higher and growing compared to the samples of treated grapes, grapes treated using Gaia at a dose of 20 or 50. We then proceeded to understand whether the molds were botrytis. And we used a typical methodology, the BSM, and it's clearly botrytis. We also carried out molecular analysis to make really sure that this was indeed botrytis. I will not explain the dendrogram, but if you can read it, it is clear that the samples taken from the grapes are much closer to the botrytis profile than to botrytis fabae or pelargoni, which are non-cinerea types of botrytis. So at least partially the mold that you see on the berries is caused by botrytis cinerea. This is the development over time of the monitoring of botrytis. During the last stage, the prevalence of botrytis seems to decline, something that we didn't notice in the early characterization of the grapes. Now, this discrepancy is possibly ascribable to the fact that probably during the late stages of the drying process, the mold might have been mixed, a combination of different molds, not just botrytis. It might be mold caused by different species, and if you just test it for botrytis, you can't really see the extent of the attack, while the molds tend to develop up until the end, as you can see in the other plot, which considers all the moldy berries and not just those attacked by botrytis. Now, also visually, these were the lots with foci where mold developed in the untreated control samples, while in the treated samples, the berries were a lot healthier. You see here the focus that often develops from a very small spot, while in the treated samples, this is what you would generally see. And again, the white spots are the deposited yeast that is still viable on the berries. Moving on to the microvinification, so this is what you see, fermentation kinetics. Sometimes fermentation kinetics provides indications as to the botrytization of the grapes. You know that grapes that are moldy or develop molds tend to display a slower fermentation kinetics. And in this case, again, this is what we saw. The untreated grapes showed that their fermentation was much slower with respect to sugars, while Gaia 50 treated grapes were much faster, were fermenting much faster than the rest. We carried out other analysis, including polyphenols, another indication of the presence of botrytis on red grapes. Is very often a cause of oxidization and degradation of these compounds. What we saw here again is that during maceration, during the extraction of polyphenols from the skins and from the pips, the samples treated with Gaia still had a higher final content in terms of polyphenols than the controls. We also tested the various fractions, trying to see if there were any differences between tannins, anthocyanins, and flavonoids, other flavonoids, and the results were still conducive to the same conclusions. The treated lots were still performing better. So that takes me to my conclusions. The Gaia yeast, once applied, once sprayed on the grapes, still seems to remain viable long term and throughout the entire drying period. The treated or sprayed grapes showed a lower incidence of fungal attacks that had been artificially induced. So the grapes would appear to have been better protected by Mechincovia. Mechincovia seems not to interfere with the fermentation process, and if it does so in a favorable way. The grapes in the control samples showed, displayed a slightly delayed alcoholic fermentation, and also the polyphenol content seems to point to the fact that Gaia-treated grapes seem to behave better. Other analyses were carried out that are not shown here. From a sensory standpoint, and other analyses were carried out on the final wine that provide other indications, albeit not statistically significant, and untreated wines in particular seem to develop more of a volatile acidity than the treated samples. Again, with rotting and botrytization phenomena. Thank you very much.